In this video, we will discuss about ALDH4A1 role in the mitochondrial pyruvate import and its metabolism. So, in the first figure, they have presented a point that ALDH4 is also lies in the mitochondrial inside the mitochondria, and for that they use the staining of this TOM M20, which is the common markers for the proteins that are present in the mitochondria, and they overlap it with this ALDH4A1 signals, and we can see that merge signals, around most of these merge signals overlaps the signals for this TOMM20 and ALDH4A1, and these things they have quantified using this intensity, where most of these intensities are overlapping of this TOMM20 and ALDH4A1. And the quantification shows that around 30% of this TOMM20 and 4A1 or ALDH4A1 overlaps are there, and thereafter once they are validating that ALDH4A1 is also present in the mitochondria, then they check its impact on the pyruvate. So first they check it for this HAP3B cancer cells. So this is panel B is on the bottom side. So here they do the metabolites or metabolic analysis, volcano plots of the metabolites, where they check what are the pathways that are upregulated, and they did it for 3 different conditions. First, this knockout versus the control knockout, ALDH4A1 knockout versus control knockout, and second, reintroduction of this wild type ALDH4 versus controlled knockout, and then reintroductions of mutated ALDH4A1 and control knockout. And what they are able to see that in the case where ALDH4A1 is knocked out, they are able to see upregulations of the pyrrolines, orthogonins, arginines kind of pathways that can be seen on the positive side or in the greater than 0, whereas when they reintroduced 4A1, they see a decrease in those pathways, that is proline, orthogonine, arginines, and same things they have seen for when they introduced the mutated metabolites, when they introduce this mutated ALDH4A1 here, except for this proline, they see a decrease in the arginine, ornithines, and also for this proline, arginine, ornithines, and histidines, but proline shows a slightly on the higher side. But in general, they see a decrease in these pathways that is related to the mitochondrial pyruvate, and these things they have then quantified in this HAP3B cell lines, where for the control they see around for the control vectors they see some pyruvate signals, and for the ALDH4A1 they see a high pyruvate signals, and when this ALDH4A1 is reintroduced either in the wild type or in the mutated form, they see a decrease in this pyruvate signals. Then they check it for acetyl CoA, and acetyl CoA signals also sees that acetyl CoA, that is the TCA cycles related to they see that TCA cycle signals, acetyl CoA signals goes down when they use this ALDH4A1 knockdown, and when they reintroduced this ALDH4A1, they see some increase in these TCA cycles that can be seen in the panel F. And before moving forward, let us discuss this panel D also. So here what it is, they check the oxygen requirement when they use this, when they knock down ALDH4A1 and when they reintroduce the ALDH4A1 in the wild type or in the mutated form. And we can see that when the ALDH4A1 is knocked down, the oxygen requirement goes down. That is, the TCA cycles consumptions or the TCA cycles activities goes down. It could be the indications. And did it check it only for the mitochondrial sections? And when they reintroduce this ALDH4A1 either in the wild type or mutated form, they see that oxygen requirement again goes up. So this indicates that the ALDH4A1 has a role towards this TCA cycles also. It helps in the mitochondrial pyruvate metabolisms. So now we will go to this panel G. Panel G indicates the regular TCA cycles. What are the things that are included in the TCA cycles? So in this case, they use 13C pyruvate to check whether the other components also gets activated or how they get affected when this ALDH4A1 is knocked down or not, to confirm the impressions or the results that the scene saw in the panel D. So in order to do that, they check all these parameters in different conditions, that is when the ALDH4A1 is knocked down and when it is reintroduced in the form of wild type or in the mutated form. And they can see that when it is knocked down, they see a decrease in this other components of the TCA cycles, but when it is reintroduced either in the wild type form or in the mutated form, they see a regaining of those factors, and that is comparable to the control. They did check it for citrate, alpha, Kg, succinate, fumarate, malatate, and OAA. And they also check the glycolysis parameters, that is DHAP and PEP. They can see the glycolysis instead of TCA cycles, the glycolysis part is upregulated when the ALDH4A1 is knocked down, and which is reduced again when the ALDH4A1 is reintroduced either in the wild type form or in the mutated form. And thereafter they checked, since the glycolysis pathways are upregulated, so they checked the other parameters also, that is palmitate and glutamine. So they can see that whether this palmitate and glutamine also has an impact or role on this ALDH4A1 also has a role on palmitate or glutamine or not. In order to check that, what they did is they use this knockdown ALDH4A1 and then introduced this palmitate. So in the knockdown conditions, they can see a decrease in the oxygen requirement, but in the control conditions with palmitate, they see an increase in the oxygen requirement, and when it is knocked down, and even then when they use palmitate, they see a same signals. So this indicates that this palmitate doesn't have a significant or doesn't have a role. ALDH4A1 doesn't have a role towards this palmitate metabolisms. And for the glutamine metabolisms also, they see the same results, that is when either ALDH4A1 is present or not, when they use the high glutamines, they see a similar amount of oxygen consumptions, indicating that this ALDH4A1 doesn't have a role towards this glutamine consumptions. This all indicates that ALDH4A1 has a role towards this, not only for the mitochondrial pyruvate import, but it also has a important role to maintain this TCA cycle metabolisms or mitochondrial pyruvate related metabolisms in the mitochondria. Yeah. So that's all for this video. Thank you for your time.